Overall, DNA replication is a fairly important process. Base pairing allows each strand to serve as a pattern for a new strand, the semi-conservative replication model we discussed previously. The two strands are complementary and anti-parallel. That means that while they are matching to each other, they're in opposite directions. The phosphodiester bonds are formed by dehydration synthesis, and they connect the molecules in a 5-to-3 direction. Overall, it takes E. coli less than an hour to copy each of its 4.6 million nucleotide pairs, whereas a human cell can copy its 6 billion in only a few hours. The process is pretty accurate, with only one error per 10 billion nucleotides on average, and there are more than a dozen enzymes and other proteins that participate. Much more is known about replication in bacteria rather in eukaryotes. However, the process is fundamentally similar, so just be aware. The process of DNA replication ensures that every new cell resulting from mitotic division has identical DNA, and enzymes facilitate that process. So as we go through this, if you understand the purposes of the enzymes and the directions that things are occurring, you'll understand the most important components. For eukaryotes and prokaryotes, starting replication is a little bit different. Initiation in a prokaryote typically involves one so-called replication bubble or replicon opening. An initiator protein is going to bind to DNA and it's going to slightly unwind using ATP. That bubble will form and grow until it produces the daughter DNA. Prokaryotes, since they have a circular chromosome, they only really need one replication bubble to allow this process to occur. The eukaryotic version will typically involve multiple replicons, starting at the origin of replications, which are usually regions that are high in AT bonds, because those are weaker than GC bonds, with only two hydrogen bonds rather than three. Uh, Overall, you can see that structure here. Why would you have multiple replication bubbles? The replication is going to proceed in each direction, and you may have hundreds or thousands of origin sites per chromosome. And at the origin sites, that replication bubble will form and will elongate and will eventually fuse together. That does mean that there is some security. If a bubble fails, other bubbles around it can fill in the gaps. But you can see what those bubbles look like in the picture on the bottom right. Has been initiated, a whole host of enzymes begin to do their job. Helicase simulates the separation of the two strands, unwinding the DNA at the origin. Topoisomerase breaks and untwists by cutting phosphodiester bonds, untwisting, relieving the strain of the process. Single-stranded binding proteins prevent reannealing or prevent reattachment, so the DNA doesn't just collapse back on the replication bubble. RNA primase is going to create a starting place, an RNA primer. DNA is a double-stranded molecule and can only be extended through the DNA polymerases from a double-stranded structure. So that RNA polymerase creates a beginning site for DNA synthesis. We can see overall that looks like this. So once the actual gap is open, DNA polymerases will come in. The nucleotide will come in carrying extra phosphate groups. Those will be removed to power this bond, and it will always extend from a 5-to-3 direction. The energy is on the 5. The OH, the open bond for dehydration synthesis, is on 3. So DNA grows from 5 to 3 through this process and will begin to elongate. Now, the actual process is different depending upon direction. DNA grows from 5 to 3. So if it's building from 5 to 3, it's going to work a little bit easier, and we call that the leading strand. So in the leading strand, the new bases are forming in a 5 to 3 direction. So we can see that's going on up here. The leading strand will be 5 to 3 and will elongate continuously using that process. DNA polymerase 3 is our main DNA builder with about 1,000 bases per second. But because it works so quickly, it does tend to lead to a significant number of errors, with a mistake roughly every 1 in 10,000 bases. The other strand cannot continue in this continuous process, and we call that the lagging strand. The lagging strand is going to involve DNA polymerase III and RNA primase working together to continuously create new primers and extend them. This is going to create discontinuous fragments that can then be utilized and grown. Those fragments are called Okazaki fragments for the man who discovered them. So the primase is going to come in and it's going to make a primer in the 5-3 direction. But the replication that's happening here is trying to proceed in the wrong direction. So it's working against the flow. It can only go short distances at a time, hence the Okazaki fragments. And so that is going to create these small fragments that will eventually need to be put together. And this fragmented discontinuous structure is the lagging strand. Okazaki fragments are about 1,000 to 2,000 nucleotides long and something like in E. coli. They're significantly smaller in nucleotides, only about 1 to 200 bases. And although only one primer is needed to stimulate the leading strand, each Okazaki fragment will have its own. So to summarize, at the replication fork, the leading strand is copied continuously into the fork from a single primer. The lagging strand is copied away from the fork in short segments, each requiring a new primer. So that Okazaki fragment is going to come together to make those pieces, but now we have DNA segments that are not attached, and we have RNA segments that are not going to be stable in the long run. So overall, we have other enzymes that are going to be fixing some of these things. DNA polymerase I is another polymerase, and it's going to come in and it's going to replace the RNA primers with DNA. DNA polymerase I was the first enzyme discovered, which is why it's named number 1, and it is going to be part of this process, and is also involved in the proofreading process, correcting any mistakes that polymerase III might have left behind. DNA ligase is going to come in and it's going to seal up any broken or open phosphodiester bonds, gluing all of those pieces together. If there are mistakes in the DNA replication, enzymes called nucleases will cut that out. Okay, they will then be repaired by DNA polymerase, probably DNA polymerase I, and then ligase will come in and seal the damage. So proofreading is going to be going on throughout this process while it's going. This is why there are so few mutations in eukaryotic DNA replication. The real question gets to the ends. If DNA polymerase can only extend a double-stranded structure, then we have a bit of a problem. The final primer on the 5' end is going to be not replaceable by DNA polymerase. That RNA will be left and will eventually fall off, creating an overhang, as you can see down here. Those overhangs are problematic, and that means that every time DNA replicates, it's going to get shorter, because when that segment falls off, the complementary segment will also break off. And that is one of the things that leads to genetic components of aging and the eventual failure of cell division. Now, those ends are called telomeres, and those linear chromosome issues, it's not really something that a bacteria has to deal with because there's no end to a circle. But the telomeres are almost sacrificial. They are repetitive segments in eukaryotes, that TTA, GGG, TTA, GGG, repeated hundreds and thousands of times. And the telomeric DNA acts as a buffer zone that prevents the organism's genes from being lost or damaged in the early divisions. There are ways to recover and repair this. Cancer cells, for example, turn on the enzyme telomerase, and telomerase comes in to bind and to seal up and fix that DNA extension. There are other organisms that become temporarily triple-stranded by looping that telomere in to repair it. So basically, DNA polymerase can extend anything that has at least 2 strands, whether that is something that's supported by an enzyme or by coming more than 2 strands. It is fixable. And again, the shortening of telomeres is connected with the aging process of tissues and aging in general, and is part of this sort of life cycle of the cell. Proteins that participate in DNA replication form a single large complex, a DNA replication machine. Okay, it's a whole host of proteins and enzymes working together we collectively refer to as the replosome. These things are not sort of stationary templates. The various proteins are a single large complex of proteins that are interacting with each other in Prokaryotes, it's the parts that we've been talking about. We can see the picture on the left. In eukaryotes, multiple copies may anchor to the matrix, extending this process. You also have to deal with the removal of those helicase proteins. There are very controlled steps that are happening. So while most of the enzymes that we've discussed today are prokaryotic, the eukaryotic structure is very similar. You still see polymerases, you still see those broader structures. And that's basically it. So DNA replication is an essential component. For prokaryotes, there's one replicon. For eukaryotes, there are multiple. Okay, prokaryotes have circular chromosomes, eukaryotes have linear. In prokaryotes, this occurs in the cytoplasm. In eukaryotes, this occurs in the nucleus. Okay, prokaryotes have large Okazaki fragments. Eukaryotes have small Okazaki fragments. Prokaryotes replication is faster at about 2,000 bases per second. And for us, it's slower, about 100 base pairs per second. Why so much shorter in that process, or why so much less time? Well, we do proofreading and eukaryotes don't, which is one reason why they mutate so much more. Okay, but all of these structures are happening together, connected together in this replication machine that allows for a high fidelity passage of genetic information from the parent to the offspring.